Okay, so hola, hi all. Thank you to the organizers for inviting me to this conference one year more. It's a pleasure to be here today and to share with you the manufacturing for clinical readiness. So I am Cristina Martin. I'm the project manager in charge of CTNNV1 project at Viralgen. Viralgen is the manufacturer company that has produced the viral vector for CTNNV1. And today I'm going to present you when, how, the timelines, and the results that we have obtained for this viral vector manufacturing. Okay, so the first question is where? We are located in San Sebastián, Donosti. Maybe most of you know the city. Is 1 hour from here, and these are our facilities. So on the left-hand side of the slide, you can see the clinical site where CTNNV1 product, Viralgen, was manufactured, and then we have another site for commercialization. But if we zoom in this clinical site, you will see the installations. So here in the right part of the slide, you can see the bioreactors that we are using for manufacturing Viralgen, and we have 13 different GMP suites in which we manufacture these AAV products. So just to give you a little bit of context about why we are here today, the project started in 2017, and along these years we were able to get the clinical and the commercial certification, and our team has grown from 7 people to more than 400 along these years to make therapies like this one possible. So I also wanted to highlight our experience because I think it's important to let you know that we are capable of producing this kind of therapies. So we were working with more than 60 different clients. We have manufactured more than 1,500 batches. Out of those, more than 100 are clinical batches. And indeed, these batches are enrolled in 36 different clinical trials. So we have a lot of experience in terms of clinical manufacturing, and we are also providing this experience and leveraging that for Viralgen's product manufacturing. So we are working with different clients. You can see some on the slide, but it's not everything about working for clients. One of the key points for us is also working with foundations. For us, it's really important to be part of this journey and to also help the community. So that's why we collaborate with different foundations such as SPG50, Phoenix Next, Columbus Children Foundation, Hannah's Hope Foundation, among others. And indeed, we like to give a lot of visibility to these foundations because for us it's also important to understand the impact that this manufacturing has on this rare disease community. So, for example, you can see on the right side of the slide an event that we were organizing. The people that you see in the lower part is the people from Viralgen, and then you have Stila and Javier going there and presenting this CTNNV1 journey to our team, and I can tell you that they really appreciate it and to give sense to the work that we are doing on a daily basis. So, going back to the project and to the manufacturing process per se, I wanted to give you a brief overview about how this works. So, imagine that we have a huge tank. It can go from 250 liters up to 2,000 liters, and this tank is full of cells that are floating. The cells act as mini factories, but as you know, the operators from a factory cannot produce if they don't have instructions that let them know how to manufacture. So for that, we are giving pieces of DNA that contain this information, and these pieces are called plasmids that you have on the left side of the slide. And once we give this information to the cells act as these factories, and they are able to produce the viral vector, the AAVs. But we have this molecular cocktail in which we have the cells, the media, the reagents that we have used for the manufacturing, and we need to purify the AAVs because we cannot have that on the final product. So we do a complex process in which different steps are involved. So for those that are interested on which are these steps, we conduct a clarification step, a capture step, an ultracentrifugation step, and finally a polishing step. So when everything is done and we have purified the product, then we formulate it in the formulation buffer and we fill it in vials that are the vials that we deliver to the clinic. Another question that I would like to address today, and I received last year, and I think it's very important, is why we start with a big tank of 250 liter up to 2,000 liter, and then we end up with small vials, small volumes. What is happening in the middle? So going back to the explanation that I was giving at the beginning, in this bioreactor, we have these cells, and we need to break these cells to release the viral vectors. So when we are breaking these cells, all the organelles, all the plasmid proteins, and so all the plasmid proteins and so on are released to the media, and also we have other reagents that we were using during the manufacturing process. So we need to get rid of all of these, and we are losing a lot of volume because they are not the AAVs that we want to inject in the patients. Apart from that, as you may imagine, we cannot inject in the patients big tanks of volume. We need to concentrate the product a lot. So we need to reduce from large volumes to small volumes that can be injected in the patients. So that's why we are going from huge volumes to a few milliliters or a few liters, depending on the condition. So focusing on CTNNB1, what we have done for them. So we have started with a research-grade batch that has two purposes. The first purpose is, as this is a new product that we are putting in a manufacturing platform, we need to demonstrate that we are able to manufacture this product. So we run a first batch, and then we test different quality control assays, and we ensure that we can properly manufacture it. Another purpose of this batch is to provide material for research-grade purposes, and so then the labs have enough material to run the assays that are needed. Once this is manufactured, we move to a preclinical batch that is providing material for the toxicological studies. And once this is completed, the last step is to generate a GMP material. In this case, we did a 250-liter scale, and this is the material that is going to be used for the clinical studies. So just to give also a little bit of context about timelines, we have completed all this work in 1 year. I can tell you that it was quite challenging, and we were overlapping timelines and so on. Spila can also talk about this. But we were able to finalize all the activities in this year. And well, we started in Q2 2024. And by the end of last year, we were able to finish the preclinical manufacturing. We were overlapping this manufacturing with the GMP manufacturing. It's not the standard practice, but the scope, as I have mentioned, was to reduce the timelines and to deliver this product as soon as possible because we were conscious of the rush that this project had. Another question that I wanted to address today is, what about an additional GMP manufacturing? How long will it take? So if there is no need of additional research-grade material and additional toxicological studies, we can directly jump to the GMP manufacturing because we have already tested that the product fits into our platform and it works perfectly. So if we jump directly to the GMP manufacturing, it will take between 6 and 8 months to be completed. But what about the results that we have obtained for the batches that we have manufactured for you, for CTNM1? So highlighted in gray, you can see the historical data for this same serotype, AAV9, that we have obtained for preclinical batches. The historical data for clinical batches is highlighted in brown, and then in blue and in yellow, you have the data for the toxicological and for the GMP batch, respectively. And these are data for the productivity of these batches. So as you can see, they were quite consistent between both scales, and apart from that, the productivity was really good. It was in the upper part of the margin of historical data that we normally obtain. So it's not only about productivity, as you know, it's also about quality. I don't want to bother you putting here the huge list of QCs that we run, but I wanted to group them in 4 different types. So the first type is safety. So we need to ensure that the product that we are going to inject in the patients is safe and that it has not any microbiological contamination. Then we also need to verify that what we want to produce is indeed what we are producing. So for that, we need to test identity. Then for purity, we need to ensure that we don't have impurities to a certain level that can cause any immunological response in patients. So we also test this. And finally, we test the strength or potency in which we are ensuring that indeed the viral vector works. So for Viralgen product, we tested all these attributes, and all of them were compliant. They met the acceptance criteria, and that's why we were able to confirm the batch. So these vials are already filled and are waiting to be shipped to the clinical facilities when this is confirmed. So I want to end up my presentation thanking you for the invitation. It's a pleasure to be here. As I have mentioned, all the Viralgen team is involved in this project. They know about this story, and I am sure they would like to be here today, sharing this time with you, knowing the families, knowing the scientists behind all of this. Unfortunately, it's not possible because we will fill this room and maybe another one. But I would like to thank you because it's as I have mentioned, for us, it's a really pleasure to be here and be part of this journey. And also, I would like to thank the Viralgen team because of the huge effort that I can tell you as a project manager they were doing to accommodate this project and to do their best to be able to deliver this as soon as possible and with the best quality. Thank you.